Hi everybody, my name is John Kingsbury. I lead a group called Developability and Pre-formulation Sciences within Biologics development at Sanofi. I'm very grateful for the opportunity to be able to participate in this virtual event, and I'd like to thank Eden and the conference organizers for the opportunity. So the focus of this presentation is the manufacturability assessment of biologic drugs. This is a discipline that has arisen due to the fact that not all protein-based molecules are well suited to the established manufacturing platform. So it's become essential to anticipate these challenges early on and identify and screen for molecules that are well suited to the process. So to get at this strategy, I'll give an overview of how we think about developability assessment, how manufacturability fits into that context, and a couple of requirements for predictive assays. And I'll illustrate these principles using a case study that involves the prediction of solution behavior. Developability is a word that has different meanings in different contexts. For the sake of this presentation, we'll refer to it as the mechanism that we use to select the molecule to bring into clinical development based on its CMC-related properties. This has become such an important activity because there are so many different liabilities that can be encountered along the way. These can range from poor expression characteristics to low chemical or physical stability, sensitivity to process-related stresses, poor compatibility with process components or delivery device components, and poor in vivo outcomes as well. We organize developability assessments along three main pillars: manufacturability, stability, and delivery. Essentially, all of the different liabilities that I listed in the previous slide fit into one of these three areas. So manufacturability is about the fit to the established manufacturing platform, stability is about fit to the required dosage form, and delivery is about fit to the required route of administration, including device-mediated delivery and in vivo outcomes. All of the different developability tests are arranged in different modules within these three pillars. And the end result is that all of the data are brought together into a risk assessment that's used to compare among the different pre-candidates so that an informed decision can be made about which one to bring into clinical development. The focus today is on the first pillar, manufacturability. As I said, this is about the fit to the established manufacturing process. Poor fit to process can have a big impact on the program because this requires time and resources. Customization of unit operations, this might result in trade-offs and efficiencies or the need to innovate or expand the process control. Ultimately, in terms of risk assessment, it's about longer timelines and higher cost of good. So to understand how poor fit to process can arise, we can lay out a generic process. The DS part consists of expression, clarification, capture, viral inactivation, purification, UFDF, compounding, and freezing. The DP part consists of thawing, mixing, filtration, fill and finish, and then for the sake of completeness, we'll consider the delivery aspect here as well. So poor compatibility can arise in two different ways. It could be due to some feature or attribute of the molecule that makes it incompatible with any of the particular unit operations, or it could arise by the lability of the molecule towards stresses, the stresses that are implicit in each of the unit operations. So an example of the first is a molecule that's prone to poor solution behavior. We'll expand on this later, but it can include things like high viscosity or opalescence, phase behavior, aggregation. So if we take the example of viscosity, a molecule that's prone to becoming viscous at higher concentrations, this can lead to prohibitive back pressures during UFDF, poor filterability, issues with filling, and poor injectability. Likewise, phase behavior can limit the purification process space, can cause issues with phase separation during freezing and thawing, and likewise aggregation plays out similarly. Poor compatibility can also arise through the stresses that are implicit in the process. So these can include things like redox chemistry in the bioreactor, brief acidification during capture or viral inactivation, shear stress that is involved with the pumping and UFDF. UFDF also leads to interfacial stresses, as does freezing and thawing. And there's also compatibility with the process components and delivery device components like metals in the fill set or silicone oil in syringe barrels. So clearly there are a lot of different ways that incompatibilities can arise, and it's the goal of manufacturability to anticipate these challenges and ultimately to predict the impact to the project. So one important additional element is that manufacturability, like all developability assessment, is predictive in nature, and this is due to the stage at which it occurs prior to clinical development. So here, early on in the value chain, sample quantities are or can be limiting, and there's no at-scale process knowledge. So we are left using surrogate or microscale assays or design elements of the protein itself to sort of anticipate things that would happen at a much larger scale or over a longer time frame in the drug product. So the need for predictive tests is implicit, and within those predictive tests, it's very important to have a high bar for analytical stringency, and also that these tests be validated in the sense that they're proven to be predictive of the aspects that we are anticipating. So this has become a big mandate for our group. It's one of our primary objectives is to identify promising manufacturability or developability assays, to validate these using benchmarking data sets, and then to implement in our overall developability strategy. So I'd like to illustrate this process using an example of prediction of poor solution behavior using dilute solution interaction measurements. As I mentioned earlier, poor solution behavior can have a very broad impact on the manufacturing process. In fact, it's an impact on all three pillars of developability: manufacturability, stability, and delivery. So not only is the impact broad, but also it's composed of kind of an array of seemingly very different phenomenon. These include high viscosity, opalescence phase behavior, and aggregation. So seemingly very different things. They are linked through the commonality of being related to colloidal stability. So they're all distinctly concentration-dependent phenomenon. And the hypothesis is that the molecules are interacting, and when they're driven into proximity at higher concentration, they're self-associating into networks or clusters that can resist flow under shear, or become unstable and phase separate, or even nucleate the processes that lead to irreversible aggregation. So it's essential to identify therapeutic molecules that are well behaved in order to avoid any of these complexities later on. This figure illustrates our approach. We started with a data set of 59 different internal and approved antibodies, and we generated a high concentration data set in terms of viscosity and opalescence. This is the proof of concept data set. We also characterized these in dilute solution and performed a number of calculations on the amino acid composition as well. The results here are a set of 23 molecular descriptors that are all potentially individually or in combination predictive of the high concentration data. So we generated the high concentration set and the low concentration data set, and then looked for relationships between the two. Importantly, one of the control experiments that we did was we took the sequences in our data set and made a number of calculations of the physical properties. and compared these to the corresponding calculations from the sequences that were published in the Raybould PNAS paper last year. So here there were 239 IgG sequences, which represent the sort of clinical and approved antibody landscape. So here in the panel on the right, this is the comparison for the calculated isoelectric point. And we saw a very broad overlap with all of the different metrics that we calculated. So this gave us some confidence that our data set was molecularly diverse enough to draw general conclusions from the data. So here is the high concentration proof of concept portion of the data. This is the 59 different antibodies depicted as bars with viscosity on the top, opalescence on the bottom. These are color-coded, so viscosities higher than 30 centipois are colored yellow, and opalescence greater than 12 NTU are colored brown. And then below those thresholds are colored green. So the first take-home message here is that among these 59 different antibodies, viscosity and opalescence are actually quite common, being present in about a third of the different antibodies. Secondly, the viscosity and the opalescence appear to be mutually exclusive. So at higher concentrations, the antibodies either tended to become viscous or opalescent under these conditions and not both. We believe this to be of fundamental and practical significance. So on the practical side, it means that when looking for predictors of this high concentration behavior, since these phenomena appear to be related, it will be important to account for both phenomenon in the proof of concept data set. Also, it allows us to kind of broadly categorize our antibodies as being either well behaved, being prone to viscosity, or being prone to opalescence. So this color coding here with the green, yellow, and brown is maintained throughout the rest of the slides. So this kind of complex looking figure is how we profile the predictability of each of the dilute solution or calculation, dilute solution measurements or calculations. So here each of the antibodies is given as a box, and it's color coded according to its solution behavior category. So green, well behaved, yellow, viscous, brown, opalescent. So there are 59 different columns corresponding to each of the antibodies, and there are 23 different rows, and those rows each correspond to one of the analytics, and these are grouped as being colloidal, electrostatic, conformational, hydrodynamic, or hydrophobic. So each of the analytic outcomes are rank ordered from most favorable value on the left to least favorable value on the right. So for example, the top row is measurement of the diffusion interaction parameter, KD, which is it reflects the interactions occurring in dilute solution. The leftmost box corresponds to the antibody with the highest positive KD value, so most self-repulsive, and the rightmost box corresponds to the antibody with the most negative KD value, or most self-associating antibody, and then they're rank ordered in between. So for a good predictive assay, we would expect to see a sorting of the colors with the well-behaved ones, the green ones on the left, and the brown and yellow ones on the right. And that's exactly what we see with the diffusion interaction parameter. So qualitatively speaking, KD is a very good way of identifying well-behaved antibodies. So as you go down through the rows, you can see that also the electrostatic measures have some degree of sorting, but really nothing compares to the diffusion interaction parameter. So these weak interactions in dilute solution are really singularly able to predict the high concentration phenomenon. This is also clear in these box plots here. So on the left-hand plot, these are all the subtypes mixed together, just separated by antibody subtype. Separated by solution behavior category. So on the left are the well-behaved ones, and it's clear that repulsive interactions are really a hallmark of the well-behaved antibodies with high KD values. It's also clear that there's a threshold around 20 mil per gram where we switch from being well-behaved above that value to being poorly behaved below that value. And if we take that as a quantitative threshold, we can screen within this data set with an accuracy of over 95%. So one last interesting point is that if you parse the data by subtype, as shown on the right, it's clear that IgG1s on the left-hand side and IgG2s in the middle are kind of distributed around that threshold. IgG4s, on the other hand, have two peculiar traits. So first is that they have a very high tendency to be opalescent under these conditions, so we see most of the brown color associated with the IgG4s. They also have a universally low KD value, and we've hypothesized this to be due to the fact that the IgG4 Fc under these conditions tends to be more negatively charged or acidic than the corresponding Fc for an IgG1 or IgG2. And with the variable domain being generally positively charged, this creates a charge separation across the molecule from Fab to Fc. So we are anticipating or hypothesizing that there may be very strong anisotropic charge effects that lead to self-association for the IgG4 under these conditions. And this hints also that there may be an orientation effect implicit in the opalescent microstructure. So coming back to the generic process here, it's clear that screening for molecules with good solution behavior will have a lot of positive impacts on the development value chain. But with respect to manufacturability, it will really open up the flexibility in the unit operations and certainly it would lead to stable intermediates and drug product as well. So I'd like to conclude just by restating that manufacturability is a very critical element of the overall developability strategy. It's important to avoid poor compatibility with the process, to have a streamlined or platformized process, and this has to be predicted by manufacturability assays. So these assays are predictive in nature, and this prediction should be validated when used for screening. Finally, with respect to solution behavior, this can have a very broad negative impact on the manufacturing process. So screening for dilute solution interactions is very important early on. I'd just like to end by thanking my colleagues and developability stakeholders at Sanofi, and I'd like to thank you for your attention, and I wish you all the very best of luck in cultivating your own therapeutic pipelines.
Welcome everybody. Before I start the talk, I would like to briefly introduce myself. My name is Neunstoecklin, and I am working for Bayer since about 2017 as a process engineer in the Global Manufacturing Science and Technology Department. I'm a molecular and cellular biologist by education with an advanced degree in biochemical engineering. My experience includes early and late phase process development on several biomolecules, and I'm passionately working on technology projects for continuous and integrated bioprocessing. In my current role as a global lead process engineer, I'm the process owner of a high-volume commercial therapeutic protein and support the launch preparations of one of Bayer's late-stage biomolecules. The latter is the reason why I'm here today, to talk about a technical end-to-end project review to ensure commercial viability. Before I start with the topic, I quickly wanted to introduce as well the biopharmaceutical product supply Biotech organization and its core capabilities. We have 6 manufacturing sites distributed globally through Berlin, Leverkusen, Wuppertal, Berkeley, Gabbaniate, and Shiga. 6 innovation centers in Boston, San Francisco, Berlin, Beijing, Osaka, and Singapore. We have 5 licensed biologics on the market in more than 150 countries and 17 new molecules in development. The organization consists of more than 3,000 employees with more than 25 years of pioneering experience in biotherapeutics and single-use technology. We have deep end-to-end and manufacturing experience from cell-based fermentation over purification, fill and finish, packaging, and global distribution. We do this for clinical manufacturing as well as for commercial operations. Our manufacturing infrastructure is supported by state-of-the-art quality control and quality assurance, as well as global supply chain. We obviously ensure high standards on health, environment, and safety, and continuously improve our efforts in automation. To support our pipeline and lifecycle management, we have strong research and development and MSAT capabilities. Additionally, to ensure innovation and new technology implementation, we are in the major biotech hubs like Boston and the Bay Area. Let's zoom out even a bit more and let's ask ourselves why we're actually doing this here. And I mean, why do we do this as an industry? Roche is doing now what patients need next. Pfizer innovates to bring therapies to patients that significantly improve their lives. Novartis reimagines medicine while GSK helps people to do more, feel better, live longer. And Bayer wants health for all and hunger for none. I see an overall vision that essentially boils down to the aim to help others in need. As a student, I started out to be intrinsically interested in genetics and cellular mechanisms. Later, as a scientist, I always wanted to do something never done before, innovate. Having been part of most levels of biopharmaceutical research, development, and manufacturing, I realized that each stage has different perspectives and aims. When I was in R&D, my focus and interest was mostly on the molecules and their targets, how they interact and influence each other. I didn't think too much about production process. Then during early development, it was all about getting the right cell line and to enable it for tox studies, maybe first in human. Process robustness didn't matter too much. Then during late phase development, it was still fast to clinics and launch. But I little thought on costs of goods or process yields. So finally in manufacturing, I realized the importance of process scalability, robustness, simplicity of the process, and its cost efficiency. They are important. So most of you might already realize that each function is very focused on their own goals and which partially conflict with each other. And this ultimately can lead to misalignment. This is why at Bayer we came up with a concept we call manufacturability assessment, which essentially is a technical end-to-end project review to ensure commercial viability. Throughout this presentation, I'll essentially use both terms as synonyms. I'd like to speak about the concept and the ideas we had and present this structured assessment to ensure that launch with a commercial viable production process that fits our manufacturing network. To start out, I'll speak about the elements of such a review meeting, again about our motivation and the general scope. I'll present the implemented business process, the gates and their timing, and will introduce you to the tools established for its execution. Finally, I'll present the results of a first use case and our conclusion. First, let's think about the elements of such a manufacturability assessment. So we all have internal and external policies like SOPs and health authority guidelines or higher level business manuals for R&D and manufacturing. Those go together with operational excellence initiatives and the development and use of platforms. However, what we consider most important is a proper gap analysis, which depends on certainly SME input and their proper training. And to ensure a proper gap analysis, we believe that elements like cost of goods, demand and capacity analysis, the manufacturing network, and the assessment of the production process and its analytical package are of utmost importance. Let's consider one more time the need of such a proposed holistic and end-to-end product review. We're all familiar with the rule of 10, starting that after each quality assurance level, it will cost about 10 times more in terms of time and money to correct and fix defects as in the prior stage. And we all would agree that it is way easier to fix process issues during preclinic or before proof of concept instead of doing it during process performance qualification or even during production. So additionally, we should consider again the challenge regarding the different interest of development and product supply. For the first, it's about a process to deliver fast for toxicology studies and clinics, while costs are not a priority. On the contrary, for manufacturing or product supply, the process needs to be robust and ideally even simple, with a high yielding output and an optimized cost structure. It's, so to say, speed to market versus supply of the market. So the here presented concept tries to bridge these two views and tries to bridge the challenge itself. So the reality, at least in Bayer's past, were random activities driven by singular events. We had early facility fit assessments with external consultants based on a higher management request, plus sanity checks on global demand forecast for a late phase asset, which were surprising in the results we found, or very early cost of goods assessments on an early phase molecule. So the goal and current practice is to establish more something like a business process to ensure a commercially viable production process at competitive and optimized cost. And we aim for a process that can support the demand and fit the manufacturing network, while additionally it's predictable, robust, and reliable. We want a structured assessment conducted for all assets at a defined time point during their development life cycle, with results accessible for everybody in the company. Our mission for this technical project review meeting is to consider manufacturing needs early into the process to ensure the manufacturability of our pipeline products through early interactions with development. We established an integrated gate-based approach to convert projects to products. We did this by ensuring the commercial viability of the production process, its capability to support the demand, and its fit to the manufacturing network and our platforms. While further, we also ensure that the process is predictable, robust, and reliable. We also believe that the consequent application of such an approach will foster a targeted and accelerated process development with an efficient use of resources through backwards knowledge transfer. We plan to apply it to pipeline projects and due diligence assets, essentially of all modalities. Before we jump into the how, I'd like to spend some time on what we see in and out of scope. In scope, we consider the assessment of the process and its analytical package with regards to platform fit. Check of capacity versus demand, a preliminary check for the fit into the network, cost of goods check using available forecast data, and a transparent alignment with the biotech organization on project progress, risk, and their mitigation. Out of scope would be a formal tech transfer gap assessment, as we might all know it, and the definition and or planning for process characterization studies and their readiness check, as well as the planning for process validation and process performance qualification. We would rather consider more formal and out of scope for this assessment. So at Bayer, we have a gate-based approach for the overall product development cycle based on clinics, starting from molecule finding or lead generation over preclinical candidate confirmation, start of phase 1, 2, and 3, up to the release for submission and product launch. For our manufacturability assessment, we decided that we want to be able to modulate production for phase 1, and therefore decided to conduct it before the start of phase 1. And a second, more precise assessment, we actually consider and is planned during the phase 2, but before phase 3, so we can influence the production process that will be used for phase 3 supply. During both time points, we follow the overall high-level business process by starting with the initiation of the assessment, which you see here now on the bottom. And this assessment is initiated by the key project leaders from development and manufacturing. Afterwards, a draft report would be compiled by folks from MSAT and also by the use of several tools that I'll present in the following slides. The core piece of the assessment is usually a one-day workshop with all process development and manufacturing people involved into the project, including functions from regulatory, quality, and marketing. Agenda items for such a workshop, typical agenda items for such a workshop would be based on clinical timelines, CMC deliverables, and also detailed discussion on beforehand identified gaps or unclear process aspects. Identified risks must, at this point, not necessarily be mitigated, but they should be prioritized and, if needed, carried to later milestones, for example, lifecycle management activities after launch. So overall, the thing is aimed to generate agreement and transparency on the risks by all relevant stakeholders of the workshop. So the technical review meeting is concluded finally in a report that is collaboratively written by the participants of the workshop, which with a list of recommendations on how to move forward. Then this list of recommendations will be internally published. Actually, the whole report will be internally published to have a report in a validated document management system that ensures access to the data and the results of such a workshop to the whole company. So now let's go a bit deeper into the details and how we do this, and especially into the tools that we use for the execution of the assessment. So those are, first, tools to assess cost of goods analysis, second, to assess demand and capacity analysis, a facility fit analysis or early facility fit analysis, and on position 4 and 5, the assessment of the production process itself and its associated analytical methods. So before I go into the details of the tools, it's worth mentioning that one of the most essential input parameters for the analysis is a demand forecast, usually provided by marketing. You can see here on the left side a volume-based per dose prediction for a hypothetical biomolecule through its predicted life cycle, right, over several years. At this point, it needs to be understood that the final dose of the product is not clear yet, and the dose finding is not finally conducted. Hence, we need to prepare for the full range of the possible doses. So when using the forecast based on units, we can easily transfer this into volumetric need of drug substance as presented here on the right side. The challenge at this point is that the overall range from launch to peak, considering all doses, can easily span 1 or even 2 orders of magnitude. And I think there's no need to explain to this audience that there is a big difference in running either 1,000 liter bioreactor compared to a 10,000 liter bioreactor. So being aware of the demand and the dose range, we can feed our cost of goods analysis tools based on either simple Excel files that we developed internally, or we're using more advanced tools such as Biosolve, which might be familiar within this community, from Biopharm. Both enable us to have detailed view on the cost of the whole process on a global and overall scale, but also within individual unit operations to identify cost drivers. Further, we can quite easily, quasi in silico, calculate different process scenarios by adapting values like cell culture titer, purification yield, or alternative raw materials. And this will show us different costs. This enables not only the identification of cost drivers, but also allows us to suggest possible solutions in other scenarios. With our capacity analysis tool, which is basically a model that allows to view the number of bioreactors needed on the y-axis, depending on a given demand on the x-axis. Horizontal lines indicate the capacity utilization of a given facility. So here we have a hypothetical facility with 2 times 2,000 liter bioreactors on the left, and a second one with 2 times 10,000 liter bioreactors is given on the right. With the help of the demand planning, we can then identify the years after launch when half full, or in this case even double the capacity of the facility would be reached. The model is flexible enough to allow for different facility scales and schedules, essentially the number of slots of production bioreactors per year, as well as different processes, mainly defined by the process titer. So this tool also enables a good understanding on how capable a certain facility is to support the demand of a given product. From here, we derive the minimum amount of years in the facility until a scale out or scale up would be actually needed. This allows for better selection of the initial facility and also transparent planning for potential tech transfers after the launch. Further on, the tool we call the Bioproduction Network, it's a technical database that tries to holistically capture equipment and site-specific capabilities for all buyer sites and also for some strategic partners and CMOs. So we identified single point of contacts on the site to have annual review of all the equipment that we store in the database to ensure high quality data. And also a reasonable administration overhead. Key benefits are early and fast comparison of project needs to the existing manufacturing capabilities. It enables the targeted process and platform development. Plus, it allows us to have equipment harmonization or at least equipment bridging throughout the different sites. The cross-check of the process to the network is here visualized in the onion diagram. Simply spoken, we want the small ball inside the big one. If this is not the case, we either need to change the process to move it back into the network, or enlarge the network and make the bigger ball so big that it fits the small ball. Currently, this network consists of drug substance sites, but we aim to enlarge it also with drug production sites and maybe even more. Also, we think about gap identification of the network versus the platform process that we have in place, and then maybe also the risk associated with those gaps. The analytical methods and production process assessment tool is essentially a set of questions jointly answered and developed by the development department and manufacturing. So here, to generate a holistic and an end-to-end assessment, those questions were designed and based on the ICH M4Q module, and its structure was used to structure also our set of questions. So on the one hand, those questions aim to gather information, but on the other hand, they are also designed to prompt for potential suboptimal aspects regarding commercial manufacturing, so to say, to identify gaps. In the general, the question catalog is modified and amended based on lessons learned from each project to each project, and therefore becomes longer and longer. In the end, the result of the project review is a holistic end-to-end review. It's a documented snapshot of the project status. It identifies possible gaps and major actions needed and delivers a recommendation from manufacturing to development and senior management, listing which steps to take next. This assessment results in a list of actions that need immediate attention to enable quick to market and need to be executed before the launch actually, and during development. It also lists other topics which are of less urgent nature, but important for process cost and robustness. Those will be dedicated to become lifecycle management activities. Furthermore, the exercise allows for a very transparent communication of all gaps that are identified and good collaboration, and actually therefore good collaboration between development and manufacturing. It clarifies which activities need attention and at which point in time, and facilitates actually also the handover from development to manufacturing. And last but not least, it allows early understanding of the demand, cost, and facility needs for a development project. So in the conclusion, we can go back to the purpose of this exercise: to help others in need, or more precise in the case of this assessment, to promote the interaction between departments which have different interests, and to enable, therefore, fast to market and fast to the patient. So the applied process to do so is by the use of well-established tools and a broad cross-functional review meeting, which gives enough time to discuss the whole production change. As a result, we obtain a high-quality product that meets our customer needs at a transparent and reasonable price. We achieve reduction in the overall development timeline by early identification of manufacturing issues, efficient use of resources such as personnel, facility capacity, equipment, raw materials, capex, and time. We will harmonize the development activities following common standards. Also, finally, it avoids potential complication during the handover from development to commercial operations, and it fosters the knowledge transfer and enables the organization to align on development risks and increase overall transparency. At last, it only remains to thank you all for your attention and your interest in my talk and this topic. A quick thanks also to the team who made this happen, which was essential for the success of this program. Herewith, I'm glad to take your questions. Thank you.